Hello students, we have been learning very interesting the paper second, that is plant biotechnology, from your syllabus divide BSc. Students, in the last lecture we have got idea about the genomics. In the sense, we know that actually genome is nothing but the total genetic material of the cell, that is nothing but the genome, and the study of that one, the structures and functions, all these things we have studied in the last lecture. Today in this lecture we have to get idea about the remaining part of the topic, that is genomics and proteomics. Title of the topic is there from that one, proteomics, genomics and proteomics. What is proteomics actually? Who has introduced this terminology, isn't it? Again, what are the functions or what are the applications of this recent branch of the, I will say, the science, not only the specific life science or all sort of the things, because this branch, proteomics, I have to say that the discipline of science is having a coordination with each and every, we have to say that the science and related with the many more scientific research, isn't it? So proteomics, we know that, as already I have said to you, this proteomics, actually the terminology were coined by the, means is coined by, I have to say that, the Mark Wilkins and his colleagues in 1990. So definitely actually just one decade ago, I have to say that, 10 years back, I have to say that, this terminology has been coined by the scientist, that is the Wilkinson. Term proteome and proteomics, both the terminology have been coined by that. What is a proteome? The total protein component in the cell or organism is known as a proteome of that cell or a proteome of that organism. Total content, keep in mind, total protein component in the cell of the or organism, that is known as a proteome. Now what is proteomics? It helps in understanding the structure and function of different proteins as well as protein-protein interaction of organism. My friends, you will have to keep in mind, you have learned many more things, actually most of the things which are having related with the protein itself, isn't it? Means the physiology that is related with the, I have to say that the proteins, we know that actually each and every physiological process, whatever takes place either in animal or I have to say that in the living organism, that is catalyzed by the specific molecules, that is the enzymes. All the enzymes are proteins, but all proteins are not the enzymes. That also terminology well known to you. So my friends, I have to say that the protein-protein interaction is there, isn't it? In case of causing of diseases, in case of human power, in case of each and everything of that organism that is related with the protein. So protein, I have to say that the having a lot of importance to the protein in each and every living organisms, because we know that actually, and once we understood exact the structure, exact the function of a protein, then we will come to know that actually what the physiological changes taking place or which type of a protein is responsible to make a changes in the body of the human being, or whenever any infection takes place, at that time some changes takes place in the that organism, infected organism, but which actually, which type of changes takes place, so that can be studied with the help of this one. My friends, you will have to keep in mind one thing I will tell to you. So whenever any child take a birth, isn't it, so after 20 years of the birth of that fellow, or I have to say that the at the age of that child, isn't it, after 20 or on the 25th, I have to say that the birthday of that fellow, so which type of cancer may can takes place to that fellow, which disease may can cause to that fellow after 25 years of his birth, that can be predicted by such a type of a science. Very interesting, very, I will have to tell to you, the will have to concentrate on these things. And researchers from the globe have been taking a lot of efforts to do a research on getting idea about structure of each and every protein and its functions. Again, interaction between and among the protein, that are also studied under this, I have to say that, the new discipline, new branch of the science, not the life science, I have said to you, isn't it? So that is a proteomics. So what we have learned right now, the who has coined this terminology proteomics, isn't it? Then what is proteome? Again, what is proteomics actually? This proteomics helps in understanding the structure and function of a different proteins as well as a protein-protein interaction of the organism. My friends, a minor defect in the protein structure, its functions or alteration in expression pattern can be easily detected using proteomic studies, my friends. Already I have told to you in a simple language what actually, suppose there is a minor defect in the protein structure, suppose there is a minor defect in the function of the protein, suppose there is a little bit small alteration takes place in expression of this protein, that can be easily detected using the proteomics study. So what happens actually in the proteomics study, here the scientists, here the researchers, here the fellow who wants to get idea about the proteomics branch of a science or biology or life sciences, that fellow will have to get idea about a minor defect in the protein structure, its function, again alteration expression of the pattern can be easily detected, and that things will help just to make or find out some, we have to say that the find out the difficulties and just to overcome the difficulties of that organism by getting idea about this proteomics studies. This is important to be regarded to the development and understanding various biological processes, as proteins are the more favorable target for the various drugs. We know that actually whenever there is defect, whenever there is a problem to the health or organism, to the health of a plant or any organism takes place, at that time definitely there are the changes taking place into the body, means changes taking place into the protein of that body, proteome of that body, isn't it? And many more drugs, we know that actually the getting attack or they are target of the drugs whenever enter in the body is the protein of that body, protein of that organism, protein of that cell. Now again here the little bit definition of the proteomics is given. Proteomics is the study of all the proteins produced by the cell, that is nothing but the simple definition is this: the proteomics is the study of all the proteins produced by a cell. Proteomics is the identification, analysis, and large-scale characterization of the proteome expressed by any cells, tissues, and organs under the defined conditions. My friends, at what condition actually the changes taking place into the structure, character of the protein, isn't it? And at the specific condition, which type of expressions have been given, isn't it? Which type of changes are taking place into the body due to the change in the protein. So these studies, so simple, you have to keep in mind study of all the proteins produced by the cell, that is nothing but the proteomics. But which type of study? The identification first of all the specific protein, analysis of that protein, then large scale characterization of the proteome, which all total proteins expressed by any cells, tissues, organ under the defined condition. You have to keep in mind under the defined condition. Now, objectives of the proteomics: to characterize the post-transcriptional modifications in the proteins. Post-transcriptional modification in the proteins. To prepare the 3D map of the cell indicating the exact location of the proteins. So, in our hand, we should have a 3D map of the cell that will give us idea about the exact location of the proteins. Now, why proteomics? Because we know that in case of the genomics, DNA tells what possible. What is possible? Isn't it? Genomics gives us idea. Means DNA tells what's possible. Transcription tells RNA what probably. Probably what things are there. And proteomics, proteins, what actually happens, नक्की काय घडणार आहे, that is possible by getting idea about the proteomics, by getting idea about the thorough study of a or total protein study of the organism. So my friends, why proteomics? Because actually काय घडणार आहे, नक्की काय होणार आहे, what the changes may can take place, which type of effect may can occur, which type of physiology can get change of that organism, that can be understood very well with the help of that is nothing but the proteomics. Then study of genomics is having a lot of importance because there is a DNA which tells what possibility that we will get with the help of the study of genomics. While studying the transcription process, RNA changes in the RNA transcription, what probably, again probability is there, probably what may come, but in case of the protein, actually what happens, that can be studied with the help of the proteins, isn't it? Keep in mind, this is actually a synthesis of a protein method is there. We know that actually all the blueprint is present in the DNA, which type of protein may can synthesize. So there is a possibility that get transcribed, isn't it? DNA get transcribed, or that blueprint is given to the RNA. Transcription takes, there is also probability. But the protein whatever be produced, isn't it, synthesized, that protein will have to decide which type of changes should be taken into the human being, isn't it? So you have to keep in mind here, transcription and by translation proteins are synthesized. So there are 3 steps that we know in case of the synthesis of the protein are there, isn't it? So this is the central dogma of the protein synthesis we'll have to understand, and then you will come to know the importance of a proteomics or study of this branch. Again, why proteomics? Because I'm giving a lot of stress on this word itself and getting idea, because my friends, you have to keep in mind the behavior of the gene product is difficult or impossible to predict from the gene sequence, isn't it? We know that actually total characteristics of the organisms are under the control of the genetic material, but isn't it, the product of the gene, in the sense final product, that what product may can do, that cannot we can study by getting idea about the gene sequence from which actually the protein is to be synthesized, isn't it? Now even if the gene is transcribed, its expression may be regulated at the level of the translation. My friends, we know that actually transcription means by the process of transcription DNA transcribe the information in the form of a RNA, isn't it? And RNA then translate that into the synthesis of the proteins. Translation is there. So even if the gene is transcribed, even if gene is given a blueprint to the RNA, its expression may be regulated at the level of translation, but exact expression is regulated at the translation level or at the time of a synthesis of a protein. Okay. Now types of proteomics: structural proteomics, functional proteomics. Structural proteomics, the ultimate aim of this proteomics is to build a body of a structural information that will help to predict the probable structure and potential function for almost any protein from the knowledge of its coding sequences. So structural protein means definitely there is a study of a structure of the protein is there, isn't it? Means probable predict or probable structure of the protein should be known to you under this type of a study. Then functional proteomics, definitely it refers to the use of a proteomics techniques to analyze the characteristic of a molecular protein network involved in the living cells. In the sense, this names itself, I used to say that in English there is one of the proverb, nothing is there in the name. Or Shakespeare used to say that nothing is there in the name, but he's actually that statement, he's that universal statement gets felt in case of the science itself, because most of the things are, most of means I will say that the 100% things are in the world we can find, hidden in the world, that we have to take out. So definitely structural proteomics means the study of a protein is there in case of the proteomics, but structural means the essence study of a structure of the proteins is there definitely. Functional proteomics, that is study of a total proteins of the cell. But functional proteomics, definitely the functions of the proteins are studied under this type of a study. Okay. Now third, that is a comparative genomics. Comparative word itself tells, it refers to the quantitative study of a protein expression between the sample differing by some of the variables. In the sense, we know that actually the exact expression, exact, we have to say that the changes takes place by the protein when it interact with the various types of the various type of changes, various type of expression with various type of a substances whenever the protein interacts. So that can be studied. Comparative genomics is there. Now, what is the procedure of proteomics? There are the steps of a proteomics. That is, first one, separation of protein. Step first is separation of proteins to be isolated from the cell of which we want, of the cell or of the organism of which we want to analyze, characterize. Isn't it? All these things. So that we have to first of all isolate. One-dimensional electrophoresis, two-dimensional electrophoresis, multi-dimensional HPLC methodology are used here in case of all this separation. Then analysis of proteins by using the mass spectrometry and database utilization. Now, proteome analysis consists of actually 5 steps. Proteome analysis consists of 5 steps. Sample collection, handling and storage, most important. Separation of individual proteins by two-dimensional electrophoresis. Protein characterization, identification by the mass spectrophotometry or other methods, and storage, manipulation and comparison of the data using the bioinformatics. My friends, very simple steps are there. Very, I have to say that the few steps are there. In that one, only we know that unless and until we could analyze, unless and until we could isolate or separate the proteins, we will not get that protein. Then after analysis, separation of that individual proteins is there. We separate, we have taken into consideration first of all in the isolation total proteins we have taken. We don't know actually which type of protein we have isolated. That is a group of protein. Then that group of protein we have to separate individually. Again we have to characterize that one, learn the characteristics. Again identification by the mass spectrometry or other methods. Again we have to identify all these things. Then separate protein we have to store, we have to manipulate, and again comparison of a data using the bio. We know that actually which type of protein individually we have having in our hand, that we can compare with the bioinformatics using bioinformatics. What is bioinformatics? That also we have to study in the next our topic, that is related with the bioinformatics itself, isn't it? In that bioinformatics database are there. Protein database is stored in a NCBI. And what is NCBI? That is actually nationalized, we have to say that the one of the institute is there. Nationalized institute is there, where the all scientists throughout the globe, across the globe, are working on the structure, working on the characteristics, working on the exact, we have to say that 2D 3D structures, functions of the protein, and that data is stored in the database, isn't it? The storage is there. We can suppose we are having in our hand one of the type of a protein, characteristic of the protein, then we can that compare with the storage databases of the protein, and then we have to say that take out, we can get idea about the which type of protein is there in our hand, or which type of protein we have isolated from the organism which is in our laboratory, or which we have considered to separate the study the protein of that organism. Okay, so that also we have to study. Now, importance of proteomics, my friend, that is most important things you have to always keep in mind. Protein sample identification and confirmation of a protein, that is the first we have to say that importance. Protein sample purity determination, purity is most important. Direction of the post translation modifications, then detection of amino acid substitutions, then mass fingerprint of identification of proteins, then nutrition research, to identify the unknown proteins of the interest, quantifying the protein and peptides, protein biomarkers, isn't it? So all these are the importance of a proteomics, and that proteomics will help us just to get idea about. So whenever there is a one of the new protein is in your hand, at that time we can say that we are having the specific new protein or unknown protein in our hand. Then we have to characterize, then we have to find out the structure. Then so this is possible all because of the getting idea about the proteomics or while studying the technique that is a proteomics. With the help of this, my friends, thank you very much to you. Stay safe, stay at home, take care always. Right now also we have been passing through one of the COVID-19 pandemic. Take care of your parents also, your family, your village, then you are all I have to say that the taluka, district, and take care of all these things, because right now we are passing through one of the. But no doubt there is a under control the situation. Then also we have to follow the rules and regulations of government issued by the government. That would be the make us again safe and safe. That is for our safer side. Thank you very much. See you in the next lecture. Thank you.